Cellular respiration is the process of converting glucose into usable forms of energy. The reactants of cellular respiration are glucose and oxygen, and the products are carbon dioxide, ATP, and water. Oxygen enters the cells of an animal when it diffuses into their lungs, and glucose enters the animal when they eat other animals or eat food. The first process of cellular respiration is glycolysis. The purpose of it is to split a glucose molecule into two 3-carbon molecules so it's small enough to enter the mitochondria. Glycolysis takes place in the cytoplasm of a cell. Glycolysis has a net gain of 2 ATP, NADH, and 2 pyruvic acids, which is when it divides into 2 3-carbons. And what the pyruvic acid enters the mitochondria, and when it enters the mitochondria, it loses 1 carbon dioxide and combines with a coenzyme A to form acetyl CoA, which is a 2-carbon molecule, and some of the energy converts NAD plus into NADH, and then it enters the Krebs cycle, which is shown down here. When the acetyl CoA enters the Krebs cycle, it breaks down into carbon dioxide, releasing energy and electrons, and it makes FADH2, NADH, and ATP. The NADH and FADH2 take the electrons into the electron transport chain in the membrane of the mitochondria. And in the Krebs cycle, the 2-carbon molecule is added to a 4-carbon and forms citric acid. And then, as I said, as it loses carbon and makes more electron carriers, it goes back into a 4-carbon compound and then combines again, and the cycle repeats. In the electron transport chain, as electrons go through each of the proton pumps, it attracts protons that go across the membrane, and then as it goes, electrons keep going down. They release energy and end up converting a lot of ADP into ATP, and then at the end, the final electron acceptor is oxygen, which combines with hydrogen to create water. And that's basically cellular respiration. But in case there's no oxygen, it goes through something called fermentation, which is the purpose of converting pyruvic acid back into lactic acid or alcohol in the case of yeast and bacteria. And it recycles the NADH back into NAD+, so then glycolysis can continue again.